Hello, welcome everybody to today's seminar by Professor Yousry El-Kassaby. Before we start, I would like to ask you to switch off your cell phones so there is no interruption to the talk. Also yours, Yousry, of course. First thing to do. I want to give you some information about Professor El-Kassaby. He is the Associate Dean of Graduate and Postdoctoral Studies at the Faculty of Forestry at the University of British Columbia. He's also a Professor Honoris Causa of Prague University, Czech Republic, and he is a registered professional forester. His research areas include forestry domestication, tree breeding, seed orchards genetics, seed biology, quantitative genetics, and importantly also genomics, and tree improvement delivery systems. Professor El-Kassaby has spent many years in the forest industry before joining UBC. In fact, it was 21 years that he spent in the forest industry. But he remained affiliated with the Faculty of Forestry at UBC also during that time. He became a full professor in 2005 as a senior industrial research chair. And he has a very strong reputation of being a magnificent mentor to students and postdocs in the faculty. Professor El-Kassaby is also an IUFRO office holder related to physiology and genetics, the coordinator of Division 2. And he has been also the task force coordinator, former task force coordinator for forest and genetically modified trees, and he has produced a book that has been published by FAO. And today he will talk about genomic selection, the tug-of-war between linkage disequilibrium and pedigree. So please welcome Professor El-Kassaby for this talk.
Thank you, Olga, for this nice introduction. It's nice to be here. I'm really thrilled to talk about our research on genomic selection. By the way, Richard told me to tell you where his office is. And this is the main mall, and this is the Faculty of Forestry, and Richard is on the 3rd floor. He has an ocean view office. The reason I'm going to talk about genomic selection, the momentum for genomic selection is incredible. The train has left the station with almost 350 kilometers per hour. You cannot stop the train, and everybody is doing genomic selection. And even here in Laval, that we're a bunch of really good work done by people in the university and in the CFS, and there was a pioneering work on Spruce. But as the train moves fast, we want to make sure that we do it right, and this is the intention of my talk. For what I will be doing, I'm going to really give a very fast introduction about how we do traditional breeding and where genomic selection fits in the picture. And then we all like shortcuts. I call it the silver bullet. We all want just to be cute and find something really fast. Why should we go the hard way? And then I will be talking about Fisher's infinitesimal model. From there, I will give some data on genomic selection, the basics of genomic selection, and I have a cartoon that will let you appreciate what it is all about. And then I'll talk about how we integrate it in tree breeding, and then how we improve the accuracy, or what are the drivers for prediction accuracy. And then I will give some data from our results on genomic selection in space, give an example of spruce and Douglas fir. And then also we will have a data on independent validation. And then I want to get into the nitty-gritty of the drivers of genomic selection. Is it linkage disequilibrium or pedigree? And the role of bioinformatics on that. Hopefully by that time you'll be tired, and I'm going to do the conclusion, and we will have a happy ending. Tree improvement is a very long-term endeavor, required quite a bit of finance and organizational commitment. And there are 3 actually milestones in genomic selection that you select, you breed, and test. And you keep doing it time after time. In British Columbia, let's say for a species like the Douglas fir, this cycle completes, actually you need 25 years to complete one cycle. And the way you start doing a phenotypic selection, you go in the forest and you select the trees that you like, with all the attributes, very straight, small branches, free from disease, free of insects, good wood quality. And after you select those ones, you have many of those, and those will be the parent in the breeding program. And when you put them in the breeding program, you start making what I call it mating design or structure pedigree. Certain trees will be acting as males, certain trees will be acting as females, and we get the offspring. And then we plant the offspring in our progeny trials. And we put the progeny trials on several locations in the field, and we just go and find a corner and pray, and after a few years we go and collect the data, and we analyze the data, and then we rank the parent and offspring, and we select for the second cycle. And when we select for the second cycle, it becomes actually genetic selection, because the first one is based on phenotype. We saw the trees, we like them, but we don't know if the genetics is true. Therefore, we have to do the offspring, and based on the offspring, we could evaluate the genetic merit of the parents, and we could do either two kinds of selection, either forward selection that you select from the offspring, or backward selection, you select the parents, or mix of the two. And after you do that, you just go for the second cycle, and then the best ones, they go to the seed orchard, then you get seeds and do all the good stuff with regeneration. This cycle takes 25 years. When I was actually a graduate student, I was inspired by a few papers published by Robertson, Thode, Werhahn, all those massive giants of quantitative genetics at that time, and all of them talked about measured genes. There are genes that control certain attributes, and they have a measure effect. And at that time, actually, I was looking for a silver bullet. I was a graduate student, and I did that in my thesis. And I'm not really ashamed to say that I'm the first one who've done association genetics. And that was actually done published in 1982, but I did that in my thesis. And at that time, the scientific community was a silo scientific community. The forest geneticists live together, and the plant crop breeders are together, animal breeders together, and the medical geneticists together. And even I did the association genetics, and nobody even paid attention, because the medical geneticists would never care about paper published on trees. And that time, actually, in the 70s or late 70s or early 80s, the only available marker at that time were isozymes. And I did that, and then I was trying to find a silver bullet, and I beat it to death, and I could not really find any of those markers. However, today now, we have markers coming out of our ears. We have markers by the millions, and this is actually an association genetic studies was done by Richard postdoc, and I was involved in this discussion. And here, we've done association genetics. This is 3 years, 2009, 2010, 2011, and we're looking actually at genes controlling disease resistant. And actually, we even have the validation for all those markers, and they were repeated year after year. And that was really fantastic that we managed to find genes that control disease resistant, and we did the validation year after year after year, and we get the same associations, which is actually quite good. However, the association genetics actually has some caveats. We are actually looking for a silver bullet, a marker that controls an attribute. The caveats of association genetics are many. Statistical, let's say we have 100,000 SNPs, and we repeat the analysis one at a time. Therefore, we do the analysis 100,000 times to find the SNP that associates with the trait. Every time you do that, you're stripping the SNP out of the other ones, but genes don't work in an individual SNPs. They work together in the network. Therefore, if we do that, then actually we're exaggerating the effect of those particular markers. And then we establish a statistical threshold. And the statistical threshold is this line here. Any dot above that is significant. Anything below that is insignificant. And by the way, this is poplar, and those are the 19 chromosomes of the poplar. Therefore, when you have a statistical threshold and you do analysis 100,000 times, now we have a 5% of those 100,000 times are significant by chance. Therefore, we could get what we call the false positives. The other thing also that there are false negatives, that we could have some markers here, they have an effect, but this effect wasn't really big enough to get over the hump to be significant, because we are establishing this statistical threshold. The other thing that most of those SNPs or genes control the traits, they control 1, 2, 3% of the variance of the trait. I want to remind every one of us that the association genetics was actually motivated by human geneticists. Most of the diseases that in human genetics were a defect in one gene. Therefore, if we know this gene, we could associate the gene with the disease. And this is a major effect. In complex traits, where the traits is controlled by a large number of genes, it's very difficult to do that. And we have something also called the Bemis Effect. And this Bemis Effect, that when we go and say 1, 2, 3, all those guys are significant, and we ignore the contribution of all these guys there, therefore we cancel some genes that they have effect, and we over-exaggerate the effect of the ones that are significant. Because of that, a very bright animal geneticist, his name is Yousry, actually came up with the concept of association genetics. But before I get to, genomic selection. But before I get to genomic selection, I want to just have a backdrop that we need to think about. Fisher infinitesimal model, this is 1918. This is not rocket science. And he established the principle of the infinitesimal model and say complex traits are traits that control by a large number of genes. They work while they work independently, but their effect is additive, cumulative, and they could actually affect the trait. And here's an example of complex traits. Milk production, egg production, and wood production. The difference between the egg production and the milk production, the animals are actually in a very controlled environment. Our trees actually are influenced by environment, and then the traits that we're interested in, which is mainly wood density or volume, we have to wait for 30, 40, 50 years to be able to assess those traits, for we need a long period of time, and they have low heritability because they're affected by the environment. And those are the traits that Fisher was talking about. Therefore, if we are trying to find an association genetics, which is something that follow the infinitesimal model, we need to be very lucky to find those genes that control these traits. Okay? Now, when the genomic selection came, it's a massive paradigm shift. Everybody's doing it. I'll tell you something. There is not a biological field of science where genomic selection has been implemented, even in human. But in human, it's not called genomic selection. It's called genomic prediction. They do DNA fingerprinting. They have the models, and they say your predisposition for a particular disease will be this and that. It's been implemented everywhere, but it comes with a repercussion. In the classical quantitative genetics, that we used to say the line of descent. That's why breeders make crosses, and they create the structured pedigree, and we follow the pedigree, father, mother's offspring, and all that. In the genomic selection approach, and also association genetics, the allele. If the allele is present or absence. Therefore, we could associate the presence of particular allele or group of alleles with a particular attribute. Therefore, that's a totally different mindset. My first quantitative genetics I took is with 1960s. In the 1960s, I was taught how to do quantitative genetics, and I have to have structured pedigree, half SIPs and full SIP families, so we do the quantitative genetic analysis. With the genomic selection, a line of descent is off the window. Now, do we have the allele or not? And now I have a cartoon to explain how genomic selection works. The theory of genomic selection is based on something called the linkage disequilibrium. And the linkage disequilibrium works like that. I have created my hypothetical genome. This is the genome I created, and this genome has 5 chromosomes. And then, if we believe in the infinitesimal model, therefore we have a trait, and this trait could be height, could be diameter, could be wood density, could be anything controlled by all those yellow triangles. I don't know what they are, I don't know what they do, but they are scattered through the whole genome, if you follow the infinitesimal model. Now, if I take this individual and I measure the wood density for this individual, and the wood density is so-so, and then I take this individual and I sequence it, and then I get my SNPs, those SNPs there. They also, I don't know where they are scattered throughout the whole chromosomes, and then I get into scenarios. One scenario that I have tract of chromosomes where I have SNPs, but I don't have any of those causal genes. I could have another scenarios where the SNP could be inside the gene, or could be very close to the gene. They migrate together. They will have a non-random segregation. This is what we call linkage disequilibrium. Therefore now, if this individual has a height, and I know the height is 2 meters, and now I have another data, hard data of the SNPs. Therefore I have a population of SNPs in one hand, and I have the height in another hand. I want you to imagine that we've done that on 1,000 individuals. Therefore we have a population of SNP genotyping for those 1,000, and I have the height for that, for those 1,000 individuals. I could put this in actually a model. This is just a simple linear model, a regression between X and Y, but in this case, I will have multiple Ys, multiple X's, and my X's will be the SNPs. Therefore, I could have an individual has a height, and then I have 100,000 SNPs, and then another individual, another one. I could develop a regression, will be more complicated than a straight line, but regression models are predictive models. Therefore, now I have a model that using the SNP information and the height, therefore I know now which SNPs contributing to this. I don't know what they are, and I'm not really interested to know what they are, but I know that this individual has a 1 meter height, and I know has this particular SNP profile. And I could now have a predictive model where I could go and take another individuals that I didn't measure, and I take the sequence of this individual, put it in the model, I could predict the height of this individual. It's as simple regression. If we take it from there, we could actually develop prediction models, and this is actually how the animal breeders did it. They have a population, they call it reference population, they genotyped, and they phenotype how much milk they get. They put it in a predictive model, and the predictive model, very robust, is being tested and cross-validated, and then they could go now and get some animals. Those animals are never been phenotyped, but they were genotyped. We take the genotype of those animals, we put them in the model, and then the model will tell me these 3 are good, those ones are not good, and then I take it from there, and this would be my second selection. For now, I don't have to wait till the cows start producing milk, or I don't have to wait till the tree becomes 70 or 80 years old to figure the wood density. If I have a seedling that it's 1 day old, or a germinant, and I genotype this germinant, I put it in the model will predict the wood density at age 70, which is quite neat. I don't have to really complete a cycle for 20 years to figure out what the wood density, and I have actually even to wait beyond the 20 years. With this approach is very good. Because of this power of the genomic selection and actually the logic behind it, which is this linkage disequilibrium, which is the non-random segregation between the causal genes and the markers, everybody's doing this. As I said, the train left the station, and you cannot stop it anywhere. Now, there were several models of this, how we incorporate genomic selection into breeding. This is actually one of our genome projects with Alberta. And here, this is a conventional breeding. It takes 10 years to do the breeding, 13 years to do the testing, 10 years to put the trees in the orchard to get seeds. We're talking here about almost 33 years. For now, if I do 2 cycles of conventional breeding, that's a 66 years. If I do genomic selection, it will take me 2 years to do genomic selection. People in Alberta, they want to use conventional seed orchard system to produce seeds. They're not budging. This 10 will remain the same. Therefore, I said, okay, if we still stick to this, for we have 12 years, and we go to the second generation, therefore it will allow me to get 2 cycles for less than the time required for 1 cycle of conventional breeding. And that means high turnover of generations and actually increasing the genetic gain. And if we do that, we could go to this, after 1, 2 generations, we could go now to another conventional breeding cycle, because when you do genomic selection and you breed among individuals from the genomic selection, mating is going to happen, the linkage disequilibrium is going to change, before you have to start producing a new model, so the results will be useful. The point is, you could actually save quite a bit of time if the stuff works properly. Okay? Now, in order to improve the accuracy of the genomic selection models or the predictive models, we need to actually figure a few things. the trait architecture becomes very important. If it is a simple trait, like Mendel, red and white, you put it in a good soil, you put it in a poor soil, the red will be red and the white will be white. Therefore, the environment is 0. If it is a complex trait, then we have an heritability, and heritability could be ranging from a 0 to 1. Then the linkage disequilibrium between SNPs and causal gene. In my opinion, that's a very crucial parameter to be effective in genomic selection, and I'll prove it to you in a minute. And then also the number of SNPs and genome size. I have a slide that will make this easy. The pedigree structure, I'll show you something about that, and the effective population size, and the level of relatedness between the training population and validation population. Also, I will provide you with data for that. Now, this is a slide, shows the linkage disequilibrium between rice and white spruce. By the way, this came from a paper published from here, University of Laval, but I think one most important thing here is to know this is a sulfur, and this is an outcrosser. Therefore now, this distance here, from a 0 to 50 kilo base, is equal to this distance. For now, if you look at the linkage disequilibrium here, at 50 is almost 0.4, and here it's almost close to 0. Therefore, the linkage disequilibrium in outcrossing, it decays so fast. Therefore, if you want to detect linkage disequilibrium, you need hundreds and hundreds of thousands of SNPs to saturate the genome. Therefore, the distance between the causal gene and the SNP will be short, the chances for success. I want you to just keep that in mind. Here's this slide. Actually, this is true to scale. This is the spruce genome, and this is the poplar genome. The spruce genome almost 40 times bigger than the poplar, but the number of gene systems almost identical. Therefore, now the likelihood for me to do an association genetics on poplar will be high success comparing to association genetic in white spruce, because I have a massive genome, and in order to capture the causal genes, I need to have large number of SNPs. 10,000 SNPs or 7,000 SNPs or 20,000 SNPs might not cut it. But let's see. Therefore, I want you to also picture that, the size of the genome and the number of SNPs, because in those two models here, we're talking about 40,000 genes, but 40 scale difference between the two genomes, which is the white spruce and poplar. The other thing here, in my research, when I worked on the spruce, I used the GPS. This is actually the factory that generates GPS data for me. They said, okay, this is the size of the genome. This is exactly what we sequence for you. So for this little here, the sequence fragments from the GPS is this little red circle comparing to this huge genome. For in reality, even when we are dipping into sequence and we're getting hundreds of thousands or tens of thousands of SNPs, we're still looking at a small part of the genome. And even with that, we're getting lots of missing data. Therefore, it's not actually a perfect data. But I just want you to put this in perspective. Genome is huge. We need a large number of SNPs. And I have no idea where we're going to get those. But we move along. The pedigree. This is a wheat pedigree. We could have actually 100 generations. Every one of those lines is generation. So many parents, males and females. For the effective population size in plants, so huge, as opposed to animal, where you have a bull and you have a cow, and then when we move from one generation to the other, those are the bulls. And the bulls, because of artificial insemination, they control the mating and the structure of the animal population. Therefore, you could see here the effective population size in animals very low comparing to the effective population size of plants. The other thing also, the pedigree depth for wheat and barley and rice is so deep, we have lots of information, as opposed to animals. Let's say trees now, I'm converting that to trees, we only have 1 or 2 generations of pedigree. This is as far as we went with pedigree in British Columbia. This is for Douglas fir. This is actually from Balaze's thesis. This is the plus trees. This is the F1, the progeny test. And this is actually validation population, which is F2. That's as deep as it gets in forestry. Therefore, I want you to compare this to that. This is actually quite incredible when we have wheat and corn and barley, where we have hundreds of generations. You could get 2 or 3 in corn. You could get 3 generations in 1 year. 1 generation in the US, they take the kernels or the seeds, they put it in an airplane, they go to Hawaii, another generation, and they get the seeds and they put it in an airplane, they go to South Africa. You get 3 generations in 1 year. Therefore, you have a high turnover of generations. In trees, we need 20 years to move from this to this, and another 20 years. For it's quite actually a big endeavor. Now, I'm going to show you some of the results that we obtained, and we got so excited about. And this is a study that I've done with white spruce, which is 35 years old, and this is Douglas fir, 38 years old. This is, I have 25 hapsip families that I know the mother, I don't know the father. In the Douglas fir, that full sip families, I know the father and the mother. Therefore, you could see here the heritability for wood density is almost the same, 0.4 on those 2 species. Here I have an effective population size of 94 because I only know the mother, I don't know the fathers. Here I know the father and mother, the effective population size is very small. And here I did GBS, I have 62,000 SNPs. Here we have an exome capture, we have 70,000 SNPs. In every one of those studies, we have 3 sites. And here in the spruce, we selected 40 individuals with the pedigree from 40 sites replicated over 3 sites. And in the Douglas fir, we repeated the same thing with 50 individuals per site. And we did the genomic selection, and we developed predictive model for every site. By the way, note that the predictive model for full SIP families is much higher than the half SIPs. And here you could see we are all hovering around 50%. Here we are hovering around 80%. Obviously, when you have a control pedigree, you could get better data. And then we decided to see if the predictive model on this side good for this and vice versa. And we realized when we moved from one side to the other, the predictive model crashed from 0.5 to almost 50% of that. And then when we did for the Douglas fir, actually it was very good. The G by E in the Douglas fir wasn't really high, and we have almost the same neighborhood of predictive model, predictive accuracy. And we were very happy with the Douglas fir. We weren't really happy with the spruce. And then we decided to do something even better. And instead of analyzing every site individually, we put all the sites together so we could account for genotype environmental interaction. And we could see here from 50 to 62, we improved it by 10. And here it's almost with the same neighborhood, which is 0.8. And then instead of using site 1 to predict site 2 and site 2 to predict site 3, we decided to put everything together in a Big Bang, put all the sites together, account for the G by E, and we use one to predict outside. Therefore, you could see here, we're still in the 80s. Here we are in the 50s. Therefore, actually, it's quite telling that the spruce, because of the half SIPs, because of the large effective population size, is giving lower accuracies compared to Douglas fir because it's a full SIP and a larger number of SNPs. And that's actually we got very excited. We published several papers on that, and then we start thinking. But before we did that, we wanted to make sure that we do the validation. Therefore, in order to do validation for the Douglas fir, we used so many sites. The number of individuals that we use is 25,000 individuals. So when we estimate a breeding value for an individual, we do it right. And then we have 2 validation population. This is the one that was genotyped. And you could see here, A-blob means we have pedigree. GS means we have genomic selection data. Genomic selection data. And then we put all this together in one model, and we start predicting. Therefore, here, this is an independent validation. I have a mature Douglas fir, which is 38 years old. I have a juvenile, which is a validation, which is 12. And I use 3 different models, and every model has assumptions. And this model assumes that we have infinitesimal model. This one assumes that we have a gene with measure effect. This one accounts for the pedigree, part of the pedigree and the analysis. And this is what we got from juvenile height, F1, to juvenile height, F2, we're almost 90%. Exciting. We're very happy about that. We almost hit it on the head. Independent validation with the prediction model, that's a 90%. You can't beat that. It's better than the dairy industry production model better than anything I've ever seen. For now, we said, can we now take the mature height to predict the juvenile, and we jumped down from 90 to 60s. Still good. This is actually fantastic. I don't need to wait for 20 years, actually, when I have almost 70% accuracy for making a good decision. That's all good so far, but we weren't really actually convinced. For one reason. When you have a genomic data tells us two things. It tells us about the pedigree. This is what people do when they go to court. When they go to court, they're contesting paternity, they do fingerprinting, and you say, you're the father with 100%. I don't know if you've seen Murray's show on TV, where a woman goes and say, this is the father, and then they do a DNA fingerprinting to the father and the baby and the woman, and they say, yep, you're the father with 99%. But most of the time, the shows I've watched when I was sick at home, the father wasn't the father. But that's a side issue. But when you genotype large number of markers, you're capturing the pedigree, and the pedigree is the contemporary. Contemporary means who's the father, who's the mother, who's the cousin, who's the grandfather. But the most important thing is historical pedigree. A SNP is a mutation. This mutation could be 100,000 years or 200,000 years and was passed on a piece of chromosome from one generation to the other. For the likelihood that Richard and me sharing SNPs in common because of a common ancestry is very low. He's in Canada, I am from Egypt, two different continents, but the likelihood that Richard and somebody else from Quebec sharing SNPs in common is very high because it's the same gene pool. Therefore, now you're capturing the historical pedigree as well as the contemporary pedigree, and at the top of that, you're getting the LD, which is the relationship between the causal genes and the markers. These come hand in hand, and you cannot separate them. And the only way to separate those together is to do a statistical. I want to show you actually the difference between the two. This is actually a data that shows every one of those little squares is a half-zip family. Therefore, you could see individual half-zip families are not connected with each other, and the background here is white. When you use genomic data, you could see some families were connected together because they come from one stand, and then you could see here the background is a little bit darker because this is the historical pedigree. If I do a close-up, you could see this family here has 6 individuals, and those individuals are only common among the family, but these families are not connected with the other families. Every family is a standalone. When you use genomic data, those families now start connecting within a population. Therefore, and then this is the historical pedigree, and then even this with that based on the heat map here. Therefore, the genomic data is so powerful, and who needs actually to do a pedigree when you have very massive, powerful data like this? But anyhow, let's move forward. The pedigree is well linked with the linkage disequilibrium, and the only way to separate them together is to do something called deregression. This is here the mean, and this is the variance of every single family in the Douglas fir, and you could see there like that. If you do deregression, you're bringing all the mean to the families on a one line. Therefore, the pedigree effect is being neutralized. Because sometimes when you analyze the data, the family is the dominant. Therefore, every time you're analyzing the data, the family mean controls the analysis. When we do deregression, we took out the family effect. Therefore, but the variance is still the same within the family. And when we do the analysis, for now I have a situation where I took out the genetics, and then I could repeat the analysis based on pedigree, where there is no genomic data, and here this is genomic selection, where I have the pedigree and the LD. And if you look here, all the numbers are identical. In reality, if the pedigree without the SNP data is giving similar prediction accuracy or even higher than the genomic data, that tells me that actually the predictive accuracy of all the models that we produced and published are driven by the pedigree, not the LD. Now I want to start thinking about how fast the LD decays, how big the genome, how small of a SNPs that we are getting. Therefore, now the chances that we are actually, the predictive models that we produced are driven by the LD is actually not true. It's actually driven by the pedigree. But wait. For now, I showed you that we've done independent validation. This is the independent validation. This is the results that I showed you between juvenile and juvenile, okay? And then look at this one here, juvenile and juvenile from 90s to zeros. Therefore, all the prediction that I did used genomic data, which is actually combination between the pedigree and LD. When I took the pedigree out, the LD crashed. Therefore, in reality, the genomic selection models that we are all published are driven by pedigree, not by LD. Because I want you to go back to the papers and you look at them. Some of them are 7,000 SNPs, some of them are 4,000 SNPs. Our papers, we have 70 and 80,000, and we did not capture LD because this genome is so huge. And to prove this point, here is the analysis. This is for the Douglas fir, this is for the spruce. And what we did, we start changing the number of SNPs. 10,000, 20,000, 30,000, 40,000, 50,000. This is the height, and those are the wood density. Those two straight lines here, this is the A blob, which is the pedigree. If I analyze the pedigree, here is my predictive accuracy, knowing the father and the mother for wood density, and this is for height. You can note that the Douglas fir produces higher results than the spruce, and I said that that's one has half SIPs, and the other one is full SIPs. But after I reach a plateau of 7 to 8 to 10,000 SNPs, the predictive accuracy doesn't really improve. Doesn't matter how many SNPs I add up to a certain point. Here, from a 0 to 50,000, I am not making any improvement whatsoever comparing to the pedigree. This slide proves, again, that the genomic selection is driven by pedigree, not by LD, because if it was LD, this predictive accuracy should be here, that if LD is being adding. Therefore now, when we got that, we were quite depressed, and we realized that we need to have a very good bioinformatician in the group. And we got that. Now, I want you to look at this. This is the white spruce, and every one of those here in this line here is an individual, and these are the SNPs. Every time you have a dark square, this is a missing data. Therefore, we have actually lots of data we're getting from the GPS, and we're throwing away, we're not using. Therefore, if we do a k-neighborhood imputation, this is what we get. Therefore, from this, with lots of missing data, we actually manage to capture tremendous amount of data. So we increase the number of SNPs available for the analysis. So when we do our genomic selection, we will have more SNPs at our disposal. This is one method. The other method that actually we did, SNPs work in a very mysterious way. If I have 100,000 SNPs, those SNPs, some of them are correlated because of the LD, some of them are correlated because of the physical linkage. Therefore, sometimes I could have clusters of SNPs, and I could take a representative SNP, one SNP. That could be a representative of 1,000 or 2,000 SNPs. And then I could reduce the number of SNPs in the analysis while accounting for everything. Okay? And when I do that, here is an example where the data was 106,000 SNPs. After we did this graph domination, we reduced it to 50,000. Above diagonal, we have the pedigree based on father and mother. This is based on genomic information, and you look at those two are extremely identical. In other words, that without bioinformatics, we are dead in the water, because the sequencing facilities gives us data, and the data has lots of missing information. We throw it out. We're throwing actually some very valuable stuff. Therefore, you need a very solid bioinformatician to allow us to eke out every single step from the data, or we could be creative and use this representation of SNP. Okay, after that, we nailed it on the head. And this is here the analysis. We used the Spruce data. This is what we published with. 79,000 SNPs. This is the prediction model for height, and those are the two prediction model for width density. When we increase the SNPs from 80,000 to 90,000, no improvement whatsoever. We're still at the mercy of the pedigree. But when we went to almost 380,000, you could see here we have some improvement, an improvement 10 to 15%. We're not there yet. Even with 400,000 SNPs, we're not there yet because we have an improvement of 10 or 15%. But the point is, this is just to nail it on the head, what we've been doing and the entire forest genetics community. We were publishing papers, running like crazy, very happy, we've done genomic selection, but actually we are copying what the breeders are doing. The breeders, they keep the pedigree by making who's the father and who's the mother. We kept the pedigree using genomic data. Therefore, actually, it's even better. I showed you a slide like that. Therefore, in my opinion, in order to do a genomic selection and you want to be effective, you have to get into the 600 and the 700 and the 700,000 SNPs. So that huge genome of conifers is actually being saturated, so the LD will be captured. Okay? For I'm getting tired here, so I will conclude. Genomic selection is effective if it's done properly. We have to validate. You cannot do a predictive model and do cross-validation within the population. You have to go to an independent population, has to be related, and without validation, everything you do is actually suspect. And you need to really understand, as I said, the train is out of the station. The train is running 300 kilometers per hour. We're not going to stop the train, but we want to make sure the train is running on the tracks and running properly. Therefore, we need to understand what is the underpinning, actually, assumptions and drivers behind that, and then also that we need to make a bigger investment in the genotyping. Therefore, when I go to a GPS facilities and they will tell me that I'm going to give you 60,000 SNPs, I said, no, I need 600,000 SNPs. Therefore, we need multiple enzymes, multiple cuts, and we get better data. I'm almost done. This is me when I'm not working, and those are crazy people that give me money to do the work, and those are the true, actually, soldiers that they did the work. Ilga, Yaroslav, Omnea, Belize, Fran, and Charles. And just to prove to you, this is not a snag, it's a fish. Okay? I'm ready for questions.
Any questions?
So, Yousry, I was wondering, have you tried the validation to the next generation using diregress breeding values, using the 3078,000 SNPs?
Oh, yeah.
Yeah.
This is exactly, we did the validation with this is the validation that I showed you. This is for the Douglas fir. And the Douglas fir, that where we managed to get the number of SNPs from 90 to almost 400,000. Okay. And the validation, we haven't done it yet. But no, actually, we've done it, but I don't have the numbers yet. Belize is cranking it. But this is actually data that came almost 3 days ago. We're really hot at it. But I expected the validation is not going to be a 90%, but it won't be zero. It will be better than that because we added the more SNPs that we threw in the mix, the greater the chances for LD. And when we have an LD on the top of the pedigree, then the prediction accuracy will improve. Yeah.
So if you get some LD, then that means you can skip more cycles without having to. Well,
I would do what the animal breeders did. The animal breeders, they could use 1 or 2 generations of selection using genomic selection, and then after that they do conventional breeding. Because every time you breed, you're making crosses, and the LD that you're working on would be changed. Therefore, you have to have a new model based on the new scenario of LD. Okay? Yes, Richard.
So, thank you. So would you not be better off, instead of using GBS, to use something like exon capture? 'Cause then you
Absolutely
would-
not.
Why?
Okay, yes. I knew that you're going to do that. The exome capture, you're actually getting SNPs from genes are being expressed. If they are expressed, they are under selection. And if they are under selection, you will have a reduction in diversity. Therefore, we compared that. We realized that actually that the exome capture, that sometimes the diversity is not big enough to decipher the pedigree, because it depends. If the gene is actually very significant and under selection, selection will weed it out, because selection favors the survival to the fittest. Therefore, you're going to go and you will have a reduction in diversity. Therefore, what I would do, it depends on what I'm doing. I have a colleague in our faculty, Sally Aiken, working with adaptation. She wants to see the signature of selection. With the signature of selection, you need genes to figure out which one has to do with the phenology, which has to do with the frost hardening and all that. That's a very good approach. But I am actually trying to chase infinitesimal model, not frost hardening, not flush. I want everything. And in order to get an infinitesimal model, which is complex traits scattered through the whole genome, I need to be a hog. I need to have as many SNPs as I can. And I think based on the technology that we have now and the economy, GBS is my game.
Maybe something related to Richard's questions. Conifer, a huge amount of repetitive DNA which we're transposed on. Is it worth getting rid of those sequences, either electronically or not to exon capture, but some kind of...
Exome capture is elegant because you're working with the causal genes. But as I said, when I go now, I have a tree, and then I'm going to produce primers for a causal genes. I don't know which gene control wood density. For which one? But I could tell you which genes have something to do with frost hardening. I could tell you which gene has something to do with bud set and bud burst. This is very easy trait to figure out. And very cause and effect relationship through association is already being established. But because of the repetitive sequence, you're saying that we're getting so many SNPs in areas where they are not valuable. So big deal. They are there in the model. They are just, we are carrying a dead weight. It doesn't matter. We could get even to be more elegant if we do the animal breeders are doing. Animal breeders started with a SNP that almost 900,000. Now they have 30,000 because they start weeding out the SNPs in the model that are not contributing at all. But remember that they have a full genome sequence, their genome is annotated, and therefore they could zoom on the SNPs that on the traits of interest. We're not there yet. For your information, I'm sure you know that the Spruce sequence is more than 1 million contags. Therefore, now we claim that we sequence the Spruce. No, we sequence pieces of the genome, and we don't know even how to line them up with each other. Therefore, you're in a quagmire of segments, and you're trying to sequence those ones. Therefore, now, should I wait till everything is solved? Absolutely not. We could do a better job, and I think if we're going to increase, we are doing actually now another round of GPS in Cornell for Spruce and for lodgepole pine. But we are actually, the deal that we have with Cornell, that we're aiming for 2 million SNPs, raw data. And from the 2 million SNPs, when we do the annotation and all that, and we weed them, we might end up with 700,000-800,000 usable ones. And that, we might get there, but it remains to be seen. Yeah. Okay? Is that a question, Helga?
Any
Okay.
more questions? The audience? No? Okay. Thank you, Yousry, very much for your talk. It was a pleasure as always, and thank you
Thank you.
to all of you for attending today. Thank you.